Just give me credit.
I will. Hey guys, hope you enjoyed the video. Love you.
Don't look at me.
Okay, guys, so we're gonna go ahead and start. But before we do, I want to go ahead and make sure that we kind of track where we're going with our schedule. So we are gonna finish up chapter 9 on Thursday, and then on Friday we're gonna start chapter 10, continue on, and then next Thursday we'll finish it up, and then go ahead and do our Kahoot review. Next Friday we're probably gonna go ahead and start lecture 11, which is not a part of exam 3, but we are gonna go ahead and start early. Just because I don't want to waste time, because if you can see here, the 30th, you guys won't be here because you guys will be at Texas A&M. It's only going to be the seniors. So we're going to kind of miss some stuff. So to kind of, finish it up and make sure that we have enough time for our exam and not be behind, because we only have 2.5 weeks, because November 20th, we have to have exam 4 no matter what. Okay? No matter what, because after Thanksgiving break, which is that week right there, is what? Finals will technically kind of start finals week, but what is this first week after we come back from Thanksgiving break? What do you do in lab? Do you want lab practical and exam 4 in the same week? No, so let's not do exams and those. So again, that's why we're gonna kind of separate it out. So again, again, you're kind of on a time crunch. Just so you can kind of see, so we're gonna move at our own pace, but little, sped up a little bit. Okay, so let's go ahead and look at the objective. Again, as always, read the bullet parts. We will, number one,
Discuss
the historical
development
of the structure of,
DNA,
two,
analyze
the
structure,
of DNA, and three,
understand
the process of.
DNA verification.
So we've already talked about number one. That's what we did last Thursday. We're gonna kind of finish up number one today, and then start number two and finish number two today. Okay, and then what we're gonna be doing on page 9.6 is kind of wrapping up one and two together. Number three is all we're gonna focus on Thursday. Okay. So again, just as a quick review over not everything, just some high points. When it comes to these scientists, Frederick Griffith, he was the guy that basically said, I'm going to inject non-lethal or non-virulent bacteria into mice to see what happens. And what happened? They survived. Again, non-lethal, non-virulent, they're not gonna make the mice sick and die. But the second option, I guess, or the second round of experiments, they injected lethal or virulent bacteria into the mice, and what happened to them? They died. Then the third option was they killed the lethal strain. But how did they kill it? Heat. Again, that's the same reason why we cook our food, why we boil water. We do that to kill off bacteria, to kill off viruses. But they killed it off, and then they injected it into the mice, and what happened to them? They survived. Again, just the same thing. If we have food that has been contaminated with bacteria, but if we cook it all the way through, and we eat it, we're not gonna get sick. We're not gonna die. Again, that's the whole purpose of cooking. But then they can combine those previous two. Again, the non-lethal one, and then heat killed S, which again shouldn't have killed the mice. But when you put them together, what happened to the mice? They died. And they were trying to figure out, or Frederick Griffith was trying to figure out, why did the mice die? Why? But he couldn't figure it out. It wasn't until Avery, McLeod, and McCarty, a second group of people, that came through and said, you know what? We're gonna take those mice that you injected with, both of them, we're gonna look at only this group. We're gonna ignore these, and we're only going to look at this group. They looked at that specific group, and then they removed all of the what? But how did they remove all the proteins? They used a special group of enzymes called, where does it say over here? Proteases, and then all of the mice ended up dying. So it means that proteins cause the good bacteria to turn into bad bacteria? No. Again, then they went ahead and removed all of the what? RNA. And how do they remove all the RNA? By using RNases. But then the mice still died, which proved that, again, RNA didn't cause the good bacteria to turn into bad bacteria. But then they removed all of the DNA. But how do they remove the DNA? By using DNases. But when they did it this time, all of the mice lived. So what does that tell us? That the DNA is what's causing the good bacteria to turn into bad bacteria. Okay? And then Hershey and Chase, again, by the way, when they did this, again, it says it took several more years for the scientific community to fully embrace this conclusion. Again, a lot of scientists really weren't 100% sold on the idea that DNA was the one that was causing the transformation of good bacteria into bad bacteria. Okay. Hershey and Chase, they confirmed DNA's role in heredity by saying, we want to know what's actually going inside of the bacteria or going inside of the cell to turn good bacteria into bad bacteria. We want to know what's going inside. Is it DNA or is it really protein? Because a lot of scientists really thought it was protein. Okay. Again, they wanted to know definitively if it was DNA or protein. Again, a lot of scientists really still thought that it was protein, even though we have a lot of this evidence that said, actually, it could be DNA, or most likely DNA. But what do? They use radioactive phosphorus for DNA and radioactive for protein, and they were trying again to see what was going where?
Inside.
Inside of the cell. So they only wanted to pay attention to not the fluid, but the actual pellet. They were only focusing on the pellet. If the pellet was radioactive in either group, then that's what was going inside of the cell. But they noticed that with DNA, it was radioactive. But with the protein, it was not radioactive. So they knew what was going inside of the bacteria. DNA or protein? DNA. And DNA was what was causing those good bacteria to turn into bad bacteria. It was transforming those bacteria. And then we already talked about this guy. We already talked about them. Now, this we didn't cover, right? Okay. So go ahead and read for this one for me, please. Nicholas.
In 1958, Matthew
Masterson.
Masterson and Franklin Stahl
Mhm.
conducted experiments using isotopes of nitrogen to investigate how DNA replicates. They distinguished
Mhm.
between old and new DNA strands by using density gradients centrifugation.
Mhm.
Their results confirmed the
Semi-conservative.
semi-conservative model of replication where each new DNA molecule contains one old strand and one newly synthesized strand. This finding was crucial for understanding genetic enhancements.
So again, what they were trying to prove was how does DNA actually replicate? Again, we're not talking about the structure, we're talking about how does DNA replicate, which is the second and the third part of this lecture. But when we look at it though, they didn't know if it was a semi-conservative, conservative, or dispersive. Semi-conservative, what do we see? One strand which is old or original, and one strand is new. While conservative, one whole molecule is what? New, while one whole molecule is old. And in dispersive, we have chunks of new and chunks of old. Again, that's what they were trying to prove. But how did they do it? They use, and if you see right here, we're not gonna watch the whole video, but they basically use radioactive or isotopes of nitrogen to basically figure it out. Again, I'm gonna kind of show you this image. Right there. So again, what they were trying to figure out, was it conservative, was it semi-conservative, or was it dispersive? And what they did was, they basically fed a whole bunch of E. coli heavy nitrogen as an isotope, and then they kind of like tracked it to see kind of like where it went. Again, we're not looking exactly at all the details of the experiment. We just need to know what was the conclusion of this experiment. They used nitrogen, radioactive nitrogen, to find out that DNA replicates in what kind of manner. What is the title of the section? Semi-conservative. That means that every DNA molecule that is made is actually made up of one old strand and one what? New strand. One strand that was completely 100% newly synthesized. So I want you guys to go ahead and look at questions number 1 through 11 on page 9.2. And only question number 13, this one right here on question number 13 on page 9.6. So if you finish page 9.2, look at this one on page 9.6, okay? And if you finish this one, then do the rest of the question. And Nicholas, I would just get a piece of paper or something and just kind of like work on it so that way you can, you know, get answers and just spell that out. Do you want a piece of paper or are you gonna use it? Okay. For this first, this one? I think I meant to select neurotransmitter.
Okay.
I think that's what it was.
So just ignore that one?
Yeah, it was the one. I guess not. Why do you always get into this?
I get it from Waylon. He works there, that's why he just made this kind.
You cancer?
No, he can't do something about that.
Okay.
So it's interesting.
Okay, so let's go ahead and look at some of these questions really quickly. So number one says, what did Friedrich Miescher call the substance he isolated from the nuclei of white blood cells? He called it nuclein. Number two, what phenomenon did Frederick Griffith discover while experimenting with bacteria?
Transformation.
Which again is including or uptaking DNA and using it. Uptaking DNA and actually using it. So in this case, turning from good bacteria into bad bacteria. Number three, Griffith's experiment conclusively proved that DNA was the genetic material. True or false? Conclusively, what does that mean? That means there was no questions asked. Were there questions asked?
Yes.
A whole bunch. So again, false. Number four, the scientific community immediately accepted Avery's conclusion that DNA was the molecule responsible for heredity.
False.
Again, the nature of science is that there's a lot of skepticism about the results of their work, okay? Number five, why did Avery's experiment using enzymes to degrade different cellular components provide strong evidence that DNA is the genetic material? Because they explicitly showed that it was not proteins and that it was not RNA. Again, a lot of scientists thought that it was probably proteins. Okay, they definitively or explicitly showed, hey, it cannot be proteins and it cannot be RNA because we ruled it out. It has to be DNA. Number 6, Hershey and Chase use bacteriophages, which are viruses that only infect bacteria, labeled with radioactive blank to trace proteins. Sulfur. And radioactive blank to trace DNA. Phosphorus. To confirm which carry genetic information. Number 7, Chargaff's rules indicate that the amount of adenine and cytosine are always equal in DNA.
False.
False, because adenine is always equal to, and thymine is, or excuse me, and cytosine is always equal to guanine. Number 8, which technique did Rosalind Franklin use to capture the famous photograph 51? X-ray diffraction, which in this case is the same as X-ray crystallography as I mentioned in the notes, okay? Number 9, Watson and Crick proposed that the structure of DNA is a double helix. Number 10, the semi-conservative model of DNA replication means that each new DNA molecule consists of one old strand and one new strand. True or false?
True.
Number 10, Messelson and Stahl use isotopes of which element to distinguish DNA from newly synthesized DNA strands? Nitrogen. Okay. Again, only number 13, the top one, top left. What are we trying to do when we have these kinds of problems? We're just trying to figure out the amount of each nitrogen base in the percentage of DNA. So if cytosine is 10%, what does that mean for guanine? It's also 10%. What about for adenine and thymine?
40%.
How did I get 40%? Because I know that this is equal to 20, I subtract by 100, and that's what? And it means 80 is divided between those two, which is 40 and 40. Just like that. Okay. Again, we only do the first one. You guys can do the rest. It was just as a way to kind of refresh up. Any questions? Okay. So we're going to go ahead and move on. So we are going to look at the structure of DNA. So I'm going to go ahead and one second, let me erase this. Okay, so when we talk about DNA, we remember we have 3 different parts, or excuse me, let me go back over here. So we have DNA is a double what? Helix. And if you notice the way that I drew DNA and the way that DNA is drawn on this image, like this one or on the image that I have, DNA is always kind of drawn with like this little side right here with these little gaps, right? That's because these right here are called the minor grooves, and these are called the major grooves, which is just the gaps in the structure. That's it. But again, we know that it's a double helix, and this right here, if we notice that it's usually kind of like that. This thick black line right here, and this one right here, these are the what? What is that word? Backbone of the DNA molecule. So if that's the backbone, what structure is it? Well, first off, what is the DNA molecule composed of? If this is the whole structure, remember they're individually, collectively called such an N. The individual nucleotides. Nucleic acid is the whole thing, but they're made up of nucleotides. And nucleotides have 3 different parts. There's the sugar ring, the phosphate, sugar ring, phosphate, and the, I want to make sure that I do it correctly. We're just gonna do this, because I spelled mine wrong earlier too. I'm putting NB for nitrogen base, okay? So I'm not gonna drop it off. But if that's the nitrogen base, this is the what again? And then this is the phosphate never ever changes because it's always gonna be phosphate, no matter if it's DNA or RNA. But if this is DNA, what is this sugar called? Deoxyribose. And RNA, what is the sugar called? Ribose. And this nitrogenous base, what is it? It can be one of 5 different versions. Which one is it? Or what are they, I should ask? What are the nitrogenous bases? Adenine is one. Thymine, guanine, cytosine, and but since this is DNA, we ignore who? Uracil, because it's only found in RNA. So, but if this is adenine, thymine, guanine, or cytosine, remember that they're also divided between something called purines and something called pyrimidines. Purines and who? But remember there was a little acronym that we had to remember which ones were which. Pure as gold. So purines are adenine N, while pyrimidines are cytosine N, but cytosine and thymine all have a Y. Y, Y, Y. Okay? They all have a Y. So again, what sugar is this? Deoxyribose. With the nitrogenous base, which can be purines but purines, what's special about purines? Again, looking at this image, what's the difference between purines and pyrimidines? Adenine and guanine are purines, cytosine and thymine are purines. What's the difference in their structure? Not the bonds, in their structure. Purines tend to be bigger or smaller, or double ring or single ring. So purines have 2 rings. Again, this isn't new. We already discussed this all the way back in chapter 3. Pyrimidines have 1 ring. Purines have 2 rings, adenine and guanine. Pyrimidines only have 1 ring, which are cytosine and thymine. Does that make sense? Again, this is just a review because we talked about this all the way in chapter 3. Okay. So let me go back. Okay. Now, in this picture, we have 2 circles. Now, this one right here is dotted lines. What kind of bonds form between the nitrogenous bases? Hydrogen bonds, because again, anytime we see a dotted or a dashed line, that tells us it's a hydrogen bond. But what kind of bond is this right here that connects one nucleotide to another nucleotide? Such as a P, but has an F sound. Phosphodiester bond. Okay. So again, this right here is one nucleotide, but to connect it to another nucleotide, I'm just gonna draw this really quick. Nitrogenous base. This bond right here, which is between one nucleotide and another one, is called a what? Phosphodiester bond. Okay. Does that make sense? Again, all of this is literally review because we've already talked about it. Okay. The only thing that's new is that this is called the backbone and the minor and major group. That's it. Okay. But we said backbone, but what is the backbone? The backbone is actually just a sugar and a phosphate. Sugar and a phosphate. The nitrogenous bases are like the rungs of the ladder and are these little dotted lines. Again, the hydrogen bonds as we already kind of shown. Okay. But in this image though, what do you notice in this nucleotide over here? What do you notice that we're doing to it in the red? We're adding numbers. We're adding numbers to be able to identify the number of carbons in the nucleotide. And this is important, especially for if we look at this image. Actually, let me go ahead. On a sticky note, if you need one, I want you guys to go ahead and write it out. I'm just gonna pass it out. Take one, pass it around. Take one, pass it around. I just want you to draw this really quickly. Alright, just draw it. You don't have to think about all that. Okay, so when I look at this, again, each one of these dots is representing an actual carbon. So this right here is carbon 1, this one is carbon 2, this one is carbon 3, this one is carbon 4, this one is carbon 5. But as you see in this image, we also have this little dot or dash, which is like a little mark, like an apostrophe. That kind of, it's called 1 prime, 2 prime, 3 prime. Okay. But on the exam, again, this is really important because we need to know which carbon is attached to what. Because this can help us identify the overall structure of DNA. So, for example, carbon 1 is always attached to the nitrogenous base, yes. 1 is always attached to what? Nitrogenous base. Number 3, though, is always attached to the, is always a phosphodiester bond, and is always connected to the other nucleotide. One more time. Number 3 is a phosphodiester bond and is always connected to another nucleotide or the previous one. Number 2 really is just sitting there not really doing anything. Number 4, same thing, it's just sitting there not really doing anything. But what is number 5 doing? It's attached to the what? To the phosphate. So which 3 carbons are actually doing something, the ones that we really need to know? 1, 3, and 5. 2 and 4, the even ones, they're just hanging out there. 1 is always attached to the what? 3 is always a phosphodiester bond and attached to another nucleotide. Number 5 is always attached to the phosphate. No, that's it. Okay. But again, why do we need to know this? 1, we need to know this for the directionality of DNA synthesis, because again, DNA is always made in the 5 to 3 prime direction. So that means it's always made in this direction, starting with the phosphate and ending with the phosphodiester bond, in this direction. But in addition to that, what do you notice about these structures over here? Does this look exactly like the one that we just drew? No, it's actually flipped, right? Like a mirror image. So for example, what is this carbon right there? 1, 2, 3, 4, or 5? Remember, 3 is always attached to the phosphate, or excuse me, the nucleotide of the previous nucleotide, and it's a phosphodiester bond. 5 is always attached to the phosphate, and 1 is always attached to the nitrogenous base. What is that one attached to? Well, the phosphate's right there. That's not attached to the phosphate directly. Again, this is the point of why being able to count them is important. So we know which one it is. It's not necessarily connected to another nucleotide. I'm just talking about this one right here. It's not 1 because 1 is always attached to the nitrogenous base. So what is this one right here on the opposite side? That one is number 1. And what is this yellow one right here? 2. What is this blue one? 3. And again, one is number 4, and this green one is number 5. Again, this is why we need to learn to count, because it helps us identify what is attached. And it helps us orient or kind of figure out what's north, or kind of like the directionality of this molecule, okay? And on the other side, what do you notice about that one? And not the new strand, but the template strand. It's what? It's upside down. So again, which one is this one then? 1. What is this one?
2.
This one is? Because it's connected to the previous nucleotide. That means that this bond right here is actually a phosphodiester bond. This one is number
4.
and then this one is number 5. Again, you notice 1 and 4 are always diagonal, or like straight across from each other, or adjacent, okay? Again, 4 doesn't do anything, 2 doesn't do anything. 2 and 4, we don't need to worry about. It's just 1, 3, and 5. Any questions? Let's move on. Again, we've already kind of talked about this, but what does DNA wrap around? Histone proteins. And we didn't really talk about this part because this part is new, but DNA, once it wraps around these histone proteins, it forms a structure called a nucleosome. But why does DNA wrap around histone proteins to form these nucleosomes? It's because DNA is actually what charge? While histone proteins are, and again, as we discussed all the way back to the beginning of chapter 2, charges that are opposite do what? And charges that are similar, again, this is really important, especially when we talk about this thing right here called gel electrophoresis. In lab, in week 12 and 13, right before your lab practical, you actually, in 12, you guys make your own gel, so you have to like stir the stuff and do everything. You make the gel, you have to set it properly. And then in week 13, you guys come back and you take DNA and you use a pipette and you have to hold it and you have to drop the DNA into the different wells. And then you take electrodes, you connect it to the machine, and then you turn it on. But what's the purpose of that? It's because this end is negative and this end is, so that means that if DNA is negatively charged, which direction is it going to want to go towards? Towards the positive end. But the bigger pieces of DNA are going to take longer to get there because they are heavier, because they're more. The lighter pieces of DNA are going to get there quicker. So again, this one would be heavier, while this one would be lighter, or a smaller piece of DNA versus a bigger piece of DNA. Again, you guys will cover that in lab, but this is just understanding why, kind of like the connection between the two. But once we get nucleosomes, those kind of wrap up a little bit, and they form chromatin. And then chromatin super condenses into what we call chromosomes. So again, chromatin is kind of already condensed, but super condensed into these structures of chromosomes. That's where we get this, which we talked about in chapter 6. Okay? Does that make sense? I don't know, it's not just you guys, it's everyone today. Y'all guys just sleepy heads. Y'all did not get enough rest or something. I don't know what's on y'all with the bed or what. But okay, so DNA right here, then we wrap it around what? DNA is negatively or positively charged? And histones are? And then they form structures called? But nucleosomes condense into structures called? They make coils, but what is it? What did I say? Nucleosomes form into structures called chromatin. I think I messed up on word. But chromatin super condenses into what? Chromosomes. And just as we talked about before, chromosomes have the centromere. It's the location, like up here. Kinetochore are the proteins, and these are, each noodle is a chromatid. But is this before or after DNA replication? I heard before, but is this before or after DNA replication? How do we remember if it was before or after? If they are conjoined. Are these conjoined?
No.
They're separate?
Yeah.
Are they separate? Do I have the space going all the way down? No, what are they? Conjoined. So that means this is before or after DNA replication? After DNA replication. Okay. And again, we already talked about chromosomes too. When it came to chromosomes, how many did humans have? 46, what about, if a cell has all 46, what do we call that kind of cell? Starts with a D or an H. Diploid or haploid. So if it has a full set, all 46, what do we call it? Diploid. If it only has half, what do we call that? Haploid. And if, what kind of cells are going to be diploid? Or, in other words, have a full set? Anything that's not a gamete, which can be sperm or egg. And if they do have, if they only have half the genetic material, what do we call them? What kind of cells? Haploid. But haploid cells, what are the examples?
Like carrots.
Start with the G, the ones you guys just
Gametes.
did.
Oh.
Gametes, which are what and what?
Sperm.
Sperm and egg. So, one more time. It's okay, guys. I don't know. It's just a rough day. It's okay. All cells can either be diploid or they can be diploid means they have a full set of chromosomes. Haploid they only have half. Diploid cells are called somatic or body cells, which are literally anything except for egg and sperm. Haploid cells are only going to be gametes or sex cells, and the only two examples are egg and sperm. Okay. In addition to that, when we talk about chromosomes, right, this part is new, but there are two kinds of chromosomes. There's autosomes, and there are sex chromosomes. Autosomes, in the case of humans, are 22 out of the 23 pair. 22 out of the 23 pair. So that means everyone in this room's autosomes are about the same. Because again, like did we ever get to the mitochondrial Eve warm up? No, not yet. So again, basically everyone is basically kind of semi-related anyways, okay? So again, 22 out of 23 pair, they're called what? Autosomes. The last pair of chromosomes are called sex chromosomes. And that's where we see right here as X or in humans. This person in particular is a male or female? Females, because human females are X, while human males are X, Y. However, is that always the case? Yes or no? No, it is not. For example, some human males have XXY. That means they have an extra X chromosome. And some females have Klinefelter syndrome, which are XXX. So again, there are different kinds of genetic sex chromosomal disorders where some people have more chromosomes than others. For example, like Klinefelter syndrome, again, XXY, actually affects 1 in 5,000 males. It actually affects people at a higher rate than most people think. But do other organisms follow the XY trend? Do they? No, what do we see in birds, like chickens? The Z and W. Again, does that mean they look like a Z or a W? No, again, this doesn't look like an X to me, so it's just how we classify it, okay? And then fruit flies, they follow the X and Y chromosome set. What about grasshoppers? They have what? They have an O, or in other words, they have an absence of an X. So if they're XX, they are female. If they only have one X, they are male. Again, it's just showing us that all species have different kinds of categorizations for chromosomes. XY is not universal. Okay? Does that make sense? Okay. Alright, let's do this, because yes, one, two, three, four, five, six, seven, eight. One. Take one, pass it all the way around. Did everyone get one? Yeah, okay, perfect. Okay, so we're gonna actually go ahead and work through some of these together, just so it's a little bit easier. And that way everyone's on the same page. Okay. Number one says fill in the complementary nitrogenous bases. So we're gonna do a couple, and then we'll move on to the next question, do a couple, move on to the next question. That way you guys can work on the rest by yourself, okay? Again, number one says fill in the complementary nitrogenous bases, one purine and one pyrimidine, and draw the correct hydrogen bond. So what do we notice about this one right here? If this is a nitrogenous base, which one is it? Adenine. And what should this one be then? Because adenine always pairs up with thymine. But how many hydrogen bonds should I draw?
Two.
Because adenine and thymine always pair up with 2 hydrogen bonds. Just like that. Again, we're gonna do one at a time, work together, and I'm gonna let you guys work on here. What about G and the next one? It says G or guanine. What should the other one be? C or cytosine. But how many hydrogen bonds should I draw between them two? 3. 1, 2, 2, and 3. Just like that. What about the one right next or below that one? What should this box, again, you don't have to show it, I'm just going to show us. What should be in that box right there? So Emily says thymine. Does it have to be thymine? It could be an adenine. What could it be? So I hear, not, it can't be yours though, because we're talking about DNA. So that's the only one it can't be. But which one is it specifically? So I hear thymine, but why thymine?
Because the sequence is already pulling adenine, thymine, ranine, cytosine, so thymine.
Technically, I could have 20 adenines right in a row, or 20 guanines in a row, or 20 thymines in a row, or 20 cytosines in a row. So why does it have to be thymine? But again, the pattern is whatever the sequence of DNA is. Again, every single species uses adenine, thymine, guanine, and cytosine, so there's has to be somewhere where they repeat. What does the size of the box have to do with it? Remember, thymine and who else? Cytosine, they're what we call pyrimidines. What does that word mean? They have one ring, so they tend to be smaller. While adenine and guanine are pure as gold, are purines, which tend to be, because they have how many rings? Does that make sense? So the size of the box matters. So again, is this thymine or is this cytosine? It does not matter. But whichever one you put, it needs to make sense. So for example, you guys say thymine, so let's put thymine. But if that's thymine, then what goes in the opposite box? It has to be adenine. Again, if I had put cytosine, then what has to go here? Guanine. But since adenine and thymine are right here, how many hydrogen bonds do I form? 2. So for these 3, you can use whichever ones you want to, but it needs to make sense. One more time. You can use whichever bases you want, but it needs to make sense. It needs to make sense. Okay. Are we good with that? Thumbs up, yes. Thumbs up, thumbs up, thumbs up. Yes. Christian, yes. Kayla, yes. Okay. So we're gonna move on to number 2. Number 2 is what? Label the 3' and 5' carbon for all of what? Sugars. 3 and 5. So I'm gonna just do this first one up here and you guys can do the rest. So right here, what should be this dot right there? 1. This one should be. This one should be. This one should be. And this one should be 5. While over here, what direction, what number do I need to start with? What is this one right here?
1.
Because it's connected to the nitrogenous base. What is this one? This one is? This one is? This one is? And the tips of the pentagons are always oxygen, so we can just kind of ignore those. Okay, again, you need to do the rest of them. I just did the first one for us. Okay. Number 3 says color the components the following colors. So all of the phosphates need to be colored what color?
Yeah.
Yellow. But what shape in this model is phosphate?
Circle.
Surface. What about the sugars? Again, in this model, what shape are all the sugars? Again, in this model, all the sugars are what shape? Pentagons, there we go. Abigail, you can go ahead and turn off the thing because we're kind of done. And all of the purines